So the first part of the sort of morning in which I'll be speaking, I'll give sort of a more high-level overview of some of the work I've done. And then the second half will be much more getting into some of the details about specific ways to access data and use the software associated with this. So as motivation for this is just a small portion of the human reference genome sequence. And of this human genome sequence, only a very small percent, about 1%, codes for proteins. And the vast majority is non-protein coding. This non-coding regions in the genome. So it's sort of an important question, understanding what's encoded in this part of the genome. And what, in particular, there's a lot of association of these non-coding parts with regulatory genomic sequences. So these are, you could think of it as stoplights, which are sequence signals that are telling proteins to be expressed in certain cell types or conditions, or genes to be expressed in order to form proteins. And what's sort of motivating a lot of interest in these non-protein coding parts of the region comes from genome-wide association studies. So these are unbiased studies to identify important locations in the genome which might be associated with disease or other phenotypic traits. So the general strategy with genome-wide association studies is you take a cohort of individuals who might be healthy and another cohort of individuals who have some type of disease, phenotype you're interested in. And then you compare their genotypes, their DNA sequence, and you try to identify places of genetic variation that are common to the people who are healthy, and then there are other places which are common to the people which have the disease. And there's been now thousands of locations across the genome associated with various types of diseases. But it's been the case that a vast majority of these locations are falling outside of the sort of well-understood protein coding regions. It's some estimates around 80%. And then, so it becomes a question, can we start interpreting these? And often, another issue with these genome-wide association studies is something called linkage disequilibrium. So sometimes it won't implicate a specific SNP as being associated with the disease, but a set of SNPs, and then you might want to use other types of information to be able to prioritize which SNP is most likely the causal one. And for that, it would be important to start to be able to better interpret the non-coding parts of the human genome. So our goal here would be to take some sequence like this, and if we saw some mutations, so you see this letter changing here, could we start interpreting, would this be an important change? In what cell types might this be relevant, and so forth. So just from reading the DNA sequence directly, this could be sort of quite a challenge to make sense of it. But suppose we had markers on top of the DNA sequence. You could think of it as like Post-it notes, indicating certain important things on top of it. And you started to see patterns of these. Like quite often you would see these two shapes, a trapezoid and lightning bolt, co-occurring together here and here and here, right on top of the DNA sequence. This could be a signal to you that there's probably something important about this pattern when you see this trapezoid and this lightning bolt together. And you might see the trapezoid alone in places. You might see a long domain of pentagons together. You might see this pattern together co-occurring. So these could be important signatures that one could potentially detect. And it turns out that biology has given us the equivalent of these Post-it note markers in the terms of epigenetic modifications. And in particular, a lot of my work is focused on a specific type of modification, histone modifications. So you can think of DNA here as being wrapped around nucleosome proteins. And these nucleosome proteins are composed of an octamer of 8 histones. So you have this, what's called this whole protein DNA complex is called chromatin. And these histone proteins are subject to chemical modifications. So these are typically often on the tails, amino acid chain, the tail of the histone proteins. And these, there's a great diversity of histone modifications. There's estimates of more than 100 different modifications. And they vary in terms of which specific histone protein the modifications associated with, and then furthermore, which specific amino acid residue in the histone modifications associated with, what type of chemical is being associated, whether it's a methylation, an acetylation, and sometimes how many occurrences of that specific chemical modification, whether you have one methyl, two methyls, or three methyls at a specific amino acid residue can be different. And then there's certain modifications which have some sort of well-known associations, and there's a certain notation here. So this is saying histone 3, lysine 4, monomethylation. This is trimethylation. This is also on the histone 3 protein, but a different amino acid residue, lysine 27. And so this, if you have this specific modification, there's a general association with enhancers or regulatory regions, but it's not sort of strictly the case, a general association. And likewise for H3K4MA3 with promoter regions. So promoters are sort of the regions at the start of the genes associated with regulation. Enhancers tend to be more distal. There's certain modifications with different types of repression, and then also some modifications that might be associated with transcribed regions. And what sort of raised a lot of the computational challenges in past number of years is a protocol called ChIP-seq, which has allowed mapping these modifications genome-wide. So the way this protocol works is you have an antibody for a histone modification. So you can think of it as a lock, a key fitting into a lock. So you have a key that's targeting a specific lock which is associated with a specific modification. So say you were going after this pentagon modification, what you would do is you would sort of freeze the proteins to the DNA, and then you have this antibody go and recognize all the places where that specific modification's present, where you have that specific modified histone, and then you extract out the DNA where that antibody is locking into that specific modified histone. And so then you extract out that DNA, and then you sequence it. So you get the string of letters of the DNA where that histone modification was present. So then once you get those DNA sequences, you can map it back to the human reference genome, and then you can get density plots showing you for different modifications now, each one of these is a different modification. This is all in one cell type. What's its relative presence at various positions along the genome? And then you can do this not just in one cell type, but multiple cell types, as I'll discuss later. And as I mentioned, there's not just like one or two histone modifications, but there's dozens of different modifications that we're able to map across the genome. So you have to have an antibody that can recognize the modification. There's been several dozen histone modifications for which we have data for. So this is a reanalysis of data in CD4, from CD4 T cells from Keiji Zhao's lab. And what had traditionally been done was there had been a focus on analyzing one mark independently. And I was interested in the question, could we sort of take all the marks you have across some location in the genome and start classifying that location in the genome into one of a finite number of what we term chromatin states. And this would be based on the combinatorial and also spatial context of the various modifications. So what we were doing is we would take data like this and then learn what are sort of a finite number of patterns and then assign each location into one state. And I'll talk more about this in the coming slides. So, but what this is enabling us to do is now we're going to learn completely de novo without any prior genomic annotations a finite number of states, and then we can assign each location to one of these states. And what this gives us is a comprehensive annotation of the human genome, not just sort of in the protein coding regions, but throughout the non-coding regions as well. So the general idea, it's getting cut off on the top. Is there a way to fix?
Just the adjustment.
They tried to fix it yesterday, and they weren't able to.
Just have to lower the
Lower what?
foot, the little foot that's holding it up. Oh,
No,
I don't.
it's in the computer. It's on the computer.
It-
And 5, 6. Ascending.
I mean, yesterday we tried changing the resolution, screen resolution, and that did not work, unfortunately. Sorry.
It's not showing on now. Which one is it?
It's scrolled there. Now it's the PowerPoint. And it's 37
Start.
from current slide. Yeah.
Thank you.
Okay. So the approach we took is we use a multivariate hidden Markov model. I'll explain sort of in coming slides some of the basics about hidden Markov models. But so the general modeling approach we have is that underlying the genome, there's different types of elements that we don't get to observe directly, but that we're interested in. Like there might be enhancer elements that can be associated with regulating genes. There could be transcribed regions and starts of genes that we might be interested in, and so forth. But the key is that we're not sort of limiting ourselves to a few classes of elements. We're going to learn in an unbiased way what are sort of the underlying states. And what we do is we pre-process the genome into 200 base pair non-overlapping intervals. And the reason we focus on 200 base pairs is it's roughly the size of a nucleosome protein in the spacing region. But this is a parameter to the method that in principle could be adjusted. And then what we do is we binarize the data. So for each 200 base pair interval, we have a binary presence or absence call for each mark in each specific 200 base pair interval. And what we do is we ask if the number of sort of sequencing reads that fall within this interval for that mark supports it being present. And what we do is we use a Poisson distribution as our background model, and then ask if the number of reads sort of exceeds some threshold of significance based on a Poisson background distribution. So after our pre-processing for each mark in each 200 base pair interval, we have a binary 1 or 0 if we're saying that mark's present in that interval. And the reason we're sort of focusing on the binarization is that it tends to lead to sort of more explicit modeling of mark combinations. It makes it sort of more difficult for the model to overfit sort of non-meaningful variations in the signal intensity, and the parameters can be very interpretable. So what we're going to model with the mission distribution is a product of independent Bernoulli random variables. So what it's saying here is that, so we have this is what we're observing. And then what we're going to say is that at some location in the genome, in each interval, we're in one of a finite number of states, these hidden states that we don't observe directly. but we want to infer which hidden state we're in. So then we're going to learn the probabilities for these hidden states, which are associated with a different, depending on which hidden state you're in, you're going to have a different probability of observing different marks. So if you're in this state here, you'll have a high probability of these 2 marks. If you're in state 2, you might have a high probability of these other marks. And then if you're in this state over here, you would have a high probability, for example, of this pentagon. And another aspect of a hidden Markov model is that it captures spatial information. So it could potentially learn that, or infer that even though you didn't observe the mark here and didn't observe a mark here, you're more likely in state 6 here just because of the neighborhood information, while here you're might be in a different state 5. So this is a sort of a simplified example of a hidden Markov model. Say we were just looking at, we had 2 states and we had a single mark. So this would be state 1 and then state 2. And what the hidden Markov model is based off of is a notion of Markov transitions. So it's saying, given your current state, your past history doesn't matter. So once you're in state 1, in this case, we have a probability of 0.8 staying in state 1, and then we would have a probability of 0.2 transitioning to state 2. While if we're in state 2, what this is indicating, we would have probability 0.99 staying in state 2, probability of 0.01 going into transitioning back to state 1 here. And then given we're in a certain state, we could have different probabilities of observing the marks. So what this is saying is, if we're in state 1, we would have a probability of 0.8 of observing the pentagon. While if we're in state 2, we would have a probability of 0.01 observing the pentagon, and then it would be 1 minus that of not observing it. So what we have here is these 200 base pair intervals, and then we have at certain intervals we have the pentagons observed, and then the question, one question we would want to be asking is the state estimation problem. Given that we've observed these pentagons here, in this location, are we more likely in state 1 or are we more likely in state 2? So if we didn't have any transition information, we would label this as 1, 1, then it would be 2 because we didn't observe the pentagon, and that tends to happen in state 2. But because of the transition information, we could potentially infer we're still in state 1 over here. And then probably at some point, we're going to transition to state 2 here. So the hidden Markov model, in the state estimation problem, you're asking, given the full observed sequence, at some specific position, I, what is the probability that we're in state J? And it turns out, I'm not going to go into the sort of the details of this, but it's a well-known way to actually how to compute this called a forward-backward algorithm, which makes use of an idea called dynamic programming, if you've seen that before. There's tutorials on this that you could read further if you're interested in knowing the sort of algorithmic details about how one goes about computing these probabilities. Yeah.
So if the genome is in state 2 and then it receives the pentagon modification, does it make it more likely to go to state 1?
So yeah, I mean, so if you observe the pentagons at certain locations, then that's going to sort of increase your belief that you're in state 1 at some specific position. And then the hidden Markov model, given the observed sequence, it's going to be sort of reasoning about both the mark at that specific location, and it'll also sort of propagate information from the neighborhood as well. So it won't be sort of strictly the case that whenever you don't see a pentagon, you're in 2, and whenever you do see it, it's in 1, but it could potentially learn 1, 1, 1, 1, 1, 1, then 2 there. Okay? And then, so what this state estimation was just giving you a probability. But sometimes you're interested in these hard assignments to say, at this location, I'm in state 1 or state 2. And there's sort of 2 general approaches. And the one I was sort of alluding to, and which is implemented in this chromHMM software, is you take at each position whichever state has the maximum probability, the maximum posterior probability. So you compute the probability you're in state 1 and state 2 at each location, and then you take that probability. There's actually an alternative way to do the state assignments, and this is by doing what's the most likely single path through the model that would support the data. And this is based on a Viterbi path and a Viterbi algorithm, and also uses dynamic programming. Though I'm not going to sort of go into that because it's not going to be used as part of the method, but I just wanted to sort of, for those who might look into this further, that there are these sort of two different ways to do it. One is what's the most likely probability, sort of considering all possible paths, and most likely state considering all possible paths, or what's the state on the single most likely path. And then the other sort of computational challenge, so I sort of started by showing you some probabilities, but in practice what we have to do is we're given just sort of a string of observations, and what we need to do is actually learn those parameters I was showing you before. So what are those transition probabilities? What are those omission probabilities? So we need to learn those from the data itself. And then there's also sort of a minor initial parameters, what's the prior that you're starting in either state. And again, this is done through a sort of well-known algorithm called Baum-Welch, which makes use of ideas of expectation maximization, for those who are familiar with those terms. If you're not, you could read more about this in that same tutorial, but I won't sort of go into the details here of the exact algorithmic steps, but just the sort of important idea is that from the observed sequences, you can estimate sort of model parameters directly, which sort of are those model parameters which maximize the probability of observing the specific sequence you did given a certain number of states in the assumptions within the hidden Markov model. So I was showing you earlier that we were doing this not just based on one mark, but multiple different marks. And so you could potentially have some situation here where along the genome here you're observing both the pentagon and the trapezoid. So you're observing two different marks, and it could generalize to any number of marks. Some locations you might observe one of the marks or none of the marks. So what we do is we have different probabilities associated with each mark in each state. And we make a sort of a key modeling assumption is that the probability that you observe a mark conditioned on the state, each mark is independent. So then what that allows us to do is just to see a certain combination of marks, we just model or multiply the probability of each mark, observing each mark independently. And then that makes it much easier to fit the model. Instead of having to sort of model each of the 2 to the n combinations when you have n marks, we just need to compute a probability for each of the marks and then multiply their probabilities together. And this is just sort of presenting this a little more formally, what we're effectively doing. We have a set of parameters, omission parameters, transition parameters, initial set of parameters, and then we have V, which is our observed modifications, which can be binary presence or absence. M is the total number of marks, and then sort of T of C is the number of bins on a certain chromosome. And what we're doing is we have, mathematically, we can express the likelihood of the observations given the model parameters. So this could be, we're multiplying over all chromosomes, we're summing over all possible hidden state paths through the model, we're multiplying all the transition probabilities, and then all the omission probabilities depending on the observations, which are binary presence or absence. And then we can attempt to maximize these probabilities. It turns out that we don't have a guarantee that we're going to get a global maximum. We only have a sort of a local maximum. And one issue is that we're not given, you have to specify a number of states when you're learning these models. So in practice, what we do is we learn models across a range of number of states, and it turns out that often it's sort of just guided by the level of biological interpretation one's interested in terms of focusing on a certain number of states. Because for one given data set, in some sense, it's not always the best number of states, because depending on sort of the resolution of interpretation, in some analyses, it could be better to use one set of number or a different number. But one thing which I'll point out is that oftentimes you could identify specific states that have very clear biological interpretations only if you have a sufficient number of states in the model. So that can be sort of one way to have sort of very defensible lower bounds on the number of states that you're interested in. And then in terms of learning models with different numbers of states, that can be done in parallel. And there's certain ways of, depending on how you initialize the parameters when you start the model search, you could end up with different models. And the software I implemented has a specific sort of default way to do this, but I won't go into details about that, but it's in the paper if you're interested in reading about that. So this is showing you, going back to that T-cell data I showed you before, the mission parameters and the transition parameters. So what you're seeing here, the rows correspond to different states, the columns different marks, and then the coloring are indicating the relative probability of the marks in the state. I'll show this a little bigger on the next slide. But I just wanted to point out that we also have transition parameters between the states, which are saying given we're in this state, what's the probability we would transition to each of the other states? And then once we have one of these models inferred, we can go across the genome and assign each location to one of, in this case, we had 51 different states. So the question then is, what do these states actually correspond to biologically? So what you're seeing here, for example, is a state which is associated with high levels of 3 histone modifications, H3K36ME3, H3K9ME3, and H4K20ME3. And so what we see is a very strong fold enrichment for locations that were assigned to the state associated with zinc finger genes. So it's a certain class of genes and places where a specific protein Cap1 binds. And it turns out that a large part of an abstract of a paper published in 2006, they described that the enrichment of these 3 specific histone modifications is associated with sort of Cap1 binding and zinc finger genes. And then the lack of certain other marks, and we're sort of more systematically showing the lack of all these other marks are associated with this signature. So this is an example by applying the appropriate sort of computational model to the high throughput sequencing data. We can discover a very specific biological concept completely de novo without any prior knowledge of the biology here. And I mean, there's other examples where some of these states could point to things that you might not have expected beforehand. So for example, this is a state where if you do an aggregation plot at the end of genes, specifically the state 27 enriches towards the end of gene transcript. So it could be some certain signature associated with transcription termination potentially. And what we had done was we had more systematically gone through each of the states and overlapped it with a variety of other types of information. These could be gene annotations, other experimental data, evolutionarily conserved elements, transcription factor binding data sets, and saw the relative enrichments of these different states for these different classes. And we started giving biological interpretations to each state and distinctions between them. So we had different classes of promoter states depending on sort of where along the promoter they are, and also associated with different expression levels. And I'll show you also in the next slide different sort of biological categories. We had different classes of transcribed regions. Then we had some active intergenic regions, and some of these are important. These can be associated with these distal regulatory elements that are turning the genes on and off potentially. That could be very hard to identify without this type of data. Repressive elements, different types of heterochromatic elements, and also different classes of repetitive elements can be associated with different types of chromatin states. And then just to illustrate that sort of the importance of this de novo approach, for example, if somebody beforehand was saying, I was just interested in promoters, and you built some supervised method just to build a predictor of promoters, you might have missed the fact that we had a number of different states that were sort of heavily enriched for promoter regions, but then if we drill into the different biological categories, there are certain states that might be associated with like embryonic development that aren't enriched in any of the other states, or T-cell activation, and this was T-cell, so we were working with, or other states like cell cycle or RNA processing. So all this was, I was sort of talking about, was data from one specific cell type. It turns out a lot of the power of this epigenomic data is that unlike the DNA sequence, which is basically static across various cell types of your body, the epigenome can be highly dynamic in the sense that the same location in different cell types could have different epigenetic marks associated with this. So this can help, in some sense, interpret why we're having, in very different cell types of your body, you share the same DNA sequence, but the epigenome could be quite different in that different regulatory elements are active in different cell types potentially. So there's a lot of information to be gained by mapping these epigenetic marks, not just in one cell type, but many different cell types. So this was part of Brad Bernstein's ENCODE production group I worked with, where what they had done was they had mapped a subset of the histone modifications I was showing you before that were sort of considered key histone modifications across, at the time, 9 human cell types, which were represented to be sort of very diverse types of cell types. So this is showing you the data we had at just one location in the genome, where we have all these different histone modifications across all these different cell types. So the different colors correspond to marks in different cell types. And what we did, so before I was showing you this modeling approach just in one cell type, in order to learn and sort of reason about multiple cell types, what we did was we conceptually applied the same modeling approach, but we concatenated different cell types as if they were additional chromosomes. So we would have learn one common model across the cell types, but then go and give cell type specific state assignments. And I'll illustrate this on the next slide. So this is showing you the specific model we focused on. It had 15 chromatin states that captured different classes of promoters, active, weak, and inactive poised, different types of enhancer regions, more acetylated, what we're characterizing as a more strong enhancer, a weaker enhancer, an insulator, distal insulator state, states associated with transcription, either strong, initiation, elongation, or weak transcription, polycomb, repressed, associated with certain histone modification, low signal regions, and then states associated with repetitive elements and copy number variation. So this is a visualization of what we're effectively doing. So this is taking one gene in 4 different cell types. And what you're seeing here in the individual rows are different input marks, and then the relative intensities showing you how much that mark is sort of present at that location in that specific cell type. And what our method is effectively doing is taking all this data and it's summarizing it into a chromatin state call. So then somebody could go into the genome browser and then quickly scan any location and then see at a glance what types of chromatin states we're assigning it to in various cell types. So you can see at this location in the human embryonic stem cell, it's associated with more of an inactive poised state, while in these 5 cell types, it's a much more sort of active state with various enhancers and active promoter and transcribed regions. So going back to the original motivation I had given you was there's a lot of interest in starting to interpret genome-wide association study data, which had often been implicating these non-coding regions without any sort of additional support for the variants. So what we had done was we had taken, I'm showing you an example here of one specific GWAS study, which was looking at erythrocyte phenotypes, which can be associated with hypertension and cardiovascular disease. And what you're seeing here, the rows correspond to those 9 cell types, and the columns, these RS numbers, correspond to different SNPs. These were independent SNPs that met a significance threshold, a genome-wide significance threshold from this study, and they were, what you're seeing here, so each of these are the lead SNPs. So you can have multiple SNPs in linkage equilibrium, but these are the sort of reported SNPs. So they're associated with some location in the genome. And then what we do is we show in each of the cell types what chromatin state that SNP was assigned to. And I'm showing you how I'm coding that in this diagram here. And then what you can see is that specifically in K562 cells, a lot of those SNPs were assigned to what we were characterizing as being in a chromatin state associated with strong enhancers. And if you're familiar with the biology of these 9 cell types, this is specifically the state, the cell type which is sort of most relevant to erythrocyte phenotypes. And we had at the time went through a number of GWASs, primarily those that were in the NHGRI GWAS catalog, just the set of sort of lead SNPs. And we asked if we looked at what we were characterizing as strong enhancer states, did we see an enrichment for those SNPs, and in particular, what cell type were those enhancers from? And in this case, we had about 10 studies just from looking at those lead SNPs where we had an association between the locations of the SNPs and the cell type specific enhancers we were calling. So we saw studies looking at the erythrocyte and hematological traits associated with K562 cell types, studies looking at various immune-related studies associated with GM12878, which is a lymphoblast cell type, which is also immune-related, which makes sense, and then studies looking at lipids and cholesterol associated with HepG2, which is a liver-related cell type, which also makes biological sense. So that was all in 9 cell types. I had collaborated with the Roadmap Epigenomics Consortium, which went now across more than 100 cell and tissue types. And what differentiated it from the ENCODE efforts was that it was primarily focused on primary cell types opposed to cell lines. So this was published a few months ago. This is showing you at some specific location in the genome across the 127 cell types, a chromatin state model that was inferred, in this case, just based on a set of 5 marks into 15 states and with a similar color coding scheme as before. Now, I just won't go into as much detail about this work, but I wanted to just sort of reference this because I'll talk about this in the second half too, is that, and this is published if you want to get a lot more of the details, was this problem of epigenomic imputation. So when I was working with the consortium, we had this situation where we had more than 100 cell or tissue types which had some coverage. And actually it was the case that at least a set of 5 histone modifications mapped in every cell type with at least one experiment. But not every mark was mapped in every cell type. And you had some cell types which had many more marks than others, and then you had some marks which were mapped fairly deeply. They could cover 50 or so cell types. but it wasn't mapped in every cell type. So what you're seeing here in those that are in blue and pink were experimentally mapped. So I was interested in the question, could we, given all the information we have about the marks, computationally infer what one of these ChIP-seq experiments should look like without actually conducting the experiments? So we're effectively doing some virtual in silico experiment, leveraging the fact that we have seen that mark in a number of other cell types, and we have other marks in that specific cell type we're interested in. So then can we use all that information to infer a signal track without actually doing the experiment? And in this chromatin state context, this can be sort of an important resource to have, because we were in this situation, if we wanted to learn a chromatin state model based on a consistent set of marks across all the cell types, we were limited to only using 5 marks if we wanted to cover all 127 cell types. If we then decide to use a 6th mark, we had to cut ourselves down to 98 cell types. And if we wanted to do 7, we would go even further using the same model. So an alternative strategy is to learn the chromatin state models directly on the imputed data. And it turns out also that, and this is a more subtle but also important point, that when we say we actually have a mark mapped in an experiment, usually it means we have 1 or 2 replicates done. But there, especially in primary cell types, there can be a lot of confounding factors. There could be the environment, the genetic background, there can be experimental variability. So often some of those factors can be greater than the sort of biological difference between any 2 samples. So when we impute a data set, what we're effectively doing is we're combining information from lots of experiments, so it could potentially be more statistically robust in places. We might miss some of the very cell type specific behavior, but overall we can capture a lot of information of sort of the important signal while filtering away a lot of potentially noisy signal. And just to give you sort of a high-level overview of our strategy for doing this, and there's public software, chromimpute, which implements this. We use 2 sort of high-level classes or features for this. One is we have other marks in that epigenome or cell type we're trying to predict. So that can inform us. So what we could potentially do is if we have mark A and B mapped in a bunch of cell types, and then in a new cell type we just have mark A mapped, we can learn how mark A and B either correlate or anti-correlate with each other, and then that would give us some information about mark B. And we can do this more generally by taking all the marks we have and constructing features of their signal level at the target position for some mark we're trying to predict and within a neighborhood around it. The other class of information we have is what we have going for us is at some specific position in the genome, we can ask how has that mark behaved in other cell types. So if we're trying to infer some mark in some cell type, we can ask in other cell types, how has that mark behaved in that specific position? And in particular, we would want to emphasize other cell types or epigenomes that we think are in some sense more similar to that epigenome that we're trying to predict for. So we can construct what are called sort of k-nearest neighbor type features. So we infer what are the nearest cell types to us based on the other marks that we do have. And what we can then do is we can build an ensemble of regression trees. So if you're familiar with, for example, random forests, it's a similar type of idea, except here we're not specifically dropping subsets of features. But what we're doing is we're applying, we're constructing trees which have splits on different features. So we select what features to split on. And then we have leaf nodes where we follow whether we're either less or greater than some feature. And then when we reach a leaf node, we have some numerical prediction. And one of the challenges here is we don't have any training data for that target mark and that target cell type we're trying to predict. So in practice, what we have to do is we train on all the other cell types for which we do have that target mark available, and then just apply it in that target cell type and average their predictions. And by averaging a large number of regression trees, we can control for a lot of bias of any one predictor. So just to give you sort of some intuition about how this performs, this is showing you in blue data the observed data, and then red is the computationally imputed data using this compendium of data, where I've randomly selected 9 200 kb regions, and then for each mark I've randomly selected one of the available cellular epigenomes. So you can see it captures a lot of the information about the experiments without having done it. It doesn't always capture everything, but it does capture a lot of the peaks. And then we can go to very fine resolution. We impute at 25 base pair resolution, except for DNA methylation, we've done at single base pair resolution. So as part of the, actually, and this is a more quantitative evaluation. So what I'm showing you here is each of these corresponds to one of the different epigenetic marks, one of these different histomodifications. And then in red is the correlation between our imputed data with the actual observed data when we're holding it out. And then we can, as a sort of a baseline, compare it to what happens if we just average that mark across all the samples we had. So that's showing you in green. And another thing is, what's the best case if we just picked one single epigenome? So sort of this is baseline is, in practice, what people will often do is if they don't have a specific mark in a cell type they're interested in, they'll go into some database and sort of infer what they think is the most relevant, your closest cell type to the one they're interested in, and use that in place of it. But this is showing you, if you use that strategy, even if you sort of made the best selection possible, which there's no guarantee you would, this imputed data still has better correlation with the actual target. And probably a lot of this is because it's combining information from lots of different experiments. And this is another sort of visualization of the information in this imputed data now relative to the observed data. So what I'm showing you here is a heat map of the correlation between different experiments. So what you see in this color coding are the categorization of different samples by the high-level biological grouping, whether the sample is related to the brain, blood and T cells, embryonic stem cells, digestive, muscle, and so forth. And then what you see here is, in yellow means those two samples are more correlated, blue less correlated. And so these are different histomodifications. This is based on the observed data, and then here this is in red based on the computationally imputed data. And then when you see white, it means we don't even have that experiment done for the observed data. And what you can see sort of visually is that there's a much more well-defined block structure here corresponding to the known biology of the different samples, which is lacking for some of these observed marks here. And then for some of these samples where there's no observed data at all, we can sort of have a correlation structure which is sort of consistent with the biology of the cell types. And we can formally quantify this based on metrics such as predicting whether two experiments are annotated to belong to the same biological grouping. How well can we predict those experiments based on ranking, based on correlation, and quantify it using metrics such as area under the ROC curve, if you're familiar with those. So this is showing you here, I think that box shouldn't be there, but this is showing you here the different rows now that we have imputed data. We can learn a chromatin state model based on a consistent set of 12 marks that we were sort of most confident in their imputation, and the fact that we've seen them in quite a number of cell types ready to train on. And then what you're seeing here are now the chromatin states we've given, and now we have an annotation of the human genome based on 127 cell and tissue types based on 12 different features classifying it into one of 25 states. So it's sort of very now information-rich situation for any location in the genome in terms of this chromatin state information. So I think we're at a good stopping point.
Yeah, thank you, Jason.
And then I'll continue.